But today I will show you how you can use the InterPro website in your research. To do so, let's first have a quick reminder about what is InterPro. So it is a resource that provides functional analysis of protein sequences. This is done by predicting protein families, the presence of domains and important sites, and it combines predictive models from several member databases, and all the information available in InterPro entries is based on literature. So let's imagine you have a protein sequence like this one. So what InterPro does is it assigns protein families which are matching usually about the full length of the protein, or domains which are shorter regions in the proteins and have a particular function, or sites which can be one or more residues, for example binding sites or conserved sites. So in this example here, each gray rectangle represents an InterPro entry, which has an InterPro entry type, an identifier, and contains one or more member databases signatures, as you can see. Now this is the annotation for one protein, but what InterPro does is that it provides its annotation for the million sequences available in the UniProt database. And this can also be used to annotate new proteins. So all the member databases which are part of the InterPro consortiums have a particular focus, which is, for example, structural or functional domains, protein families, or protein features, for example, binding sites or active sites. So in this webinar, I will first present you the InterPro homepage. Then I will show you how you can analyze a protein sequence of a known function and all the information that InterPro provides about this sequence. And I will finish by presenting you different features available on the website to analyze a particular protein, a particular domain, or different member databases signatures. So let's have a look at the home page first. Now this is accessible by the www.ebi.ac.uk/InterPro. So by doing so, you will arrive on the InterPro homepage, which is shown on the screen at the moment. So here at the top, we have a menu, which leads you to different pages. The first one is a homepage where we are at the moment. The second one is to do a search. There is 3 different types of search available on the InterPro website, which is a search by sequence, search by text, or search by domain architecture. I will come back in detail to those in this webinar. We also have a browse functionality where you can filter the different InterPro entries or different signatures based on specific criteria. We have the results sections. This is where you will be able to find the results of protein search, for example. We have release notes. This is where you can find any information about the InterPro release version and the newly integrated signatures, and for example, the statistics about the signatures from a certain member databases integrated in InterPro. We also have a download section where you can download different files pre-formatted. We have a help section where you'll be able to find an FAQ and links to different tutorials about InterPro, and an box section for more complementary information. So let's have a look at the core of this homepage. So here on the left corner, we have information about the InterPro release version currently released and the date of release. Here we have a special section we have made available to access the SARS-CoV-2 information to provide in InterPro. And below, you have quick access to the different type of search I was mentioning before. This is also accessible by clicking on the search menu at the top. I will come back to this search a bit later. Following, we have some information about all the member databases that are part of the InterPro consortium. And for each of them, we have some summary information, the release version, and the number of InterPro entries that are integrated signatures from this specific member database. On the next tab, we have some information based on the InterPro entry type, for each entry type, the number of InterPro entries corresponding, and you can click on each of them for more details. We also have a species section where you can find the information based on each organism, the number of InterPro entries, and the number of proteins. On the right side here, we have information about the latest integrated InterPro entries. And so each entry is represented by a rectangle where you have information about the InterPro entry type, the InterPro entry name, InterPro entry accession, the different signatures integrated in this entry. We also have the count of the number of protein accessions matching this particular entry, the different domain architectures, and the taxonomy matches, and the 3D structures matching this particular entry. If we go a bit lower, we have a few links to the latest blog posts posted about InterPro, where you can find complementary information. We also have quick access to different tools and libraries. For example, the InterProScan, which is the tool for sequence search. We have the link to the documentation about the InterPro API, and also a link to the Nightingale library, which is the one used for the protein sequence viewer, which I will show you in a moment. And finally, at the bottom, we have the link of latest Twitter posts. So this is all the information available on the InterPro homepage. So if we summarize, we have a top menu, which allows you to access all the different features available on the website. We have information about the latest release version and its date of release. We have a quick access to the search features. We have information about the different member database, InterPro entries, and taxonomy. We have the list of latest entries, a link to the latest blog posts, and InterPro tools and libraries, and some information about the Twitter activity. So now let's focus on, you have a protein sequence of interest, but you don't know its function or domains it contains and everything. So you want to know that. To do that, you are going to perform a sequence search in InterPro. And this is done using the InterProScan software. And so I'm now going to show you how you can do that. So what you need to know is that InterProScan is the underlying software to analyze protein and nucleic sequences against InterPro signatures. So you can perform it for a single sequence, and on the web service you have the possibility to submit up to 30 sequences at a time. If you want to analyze more than 30 sequences, you can download and install InterProScan locally, which will allow you more flexibility. So before I carry on, I just want you to have in mind the following. So an InterPro entry is an umbrella to one or more database signatures from the InterPro consortium. It has a unique name and a unique identifier, and it has been assigned an entry type, which can be a homologous superfamily, represented by the letter H, F for family, D for domain, R for repeats, or S for site. So let's imagine we have this particular protein sequence and we want to know its function. So we are going to go to the InterPro website, and on the search section, for example, you are going to search by sequence. Here you can paste your sequence. And you are going to be able to do your search. Here you can, instead of pasting your sequence, you can choose to import a FASTA file, or you can choose a protein sequence example. Then you can also select advanced options. You can select this option to create another job immediately after submitting it, if you have more than one sequence to process. And here you can select one or more databases of interest. By default, they are all selected, but you can unselect them. But for now, we're going to leave it like that. You can then click on the search button to perform the search. So here is the InterProScan search result list of jobs submitted. You can see that the last submitted job is always at the top, and you can see that your sequence is searching at the moment. Once the search has been performed, you will have a tick button which mentions that the job is finished when you hover over it. So while this is processing, you can navigate anywhere on the website, and you will have a pop-up window which will show up at the right corner here to let you know that the job has been processed. But for this webinar, I have already performed the search, and I'm going to show you now the result page. So to do so, you just click on the result link here on the left, and you will be redirected to the page. At the top, you have some summary information about the protein submitted, so the title of the sequence you've submitted, its length. You can delete the sequence, and you can see the status of the job. And just below, you have some information about the protein family membership. So in this case, our protein has been found as belonging to the breast cancer type 2 susceptibility protein family. You can click on this to access the InterPro entry page corresponding. But what is more interesting in this page is the sequence viewer, which is here. So here at the top, the gray bar represents your protein. And each track here represents a different InterPro entry type, with the InterPro entry being at the top, and all the signatures from the different member databases that are integrated in this particular entry are shown below. You can choose to collapse the track to only display the InterPro entry information. You can choose to display the domain in different colors based either on the domain relationship, which is the case at the moment, or based on the different member databases, or based on their accession. You can take a screenshot of the protein viewer. Oh, as I was mentioning before, here you can see our job has completed, and you can go directly to the result page. So on the viewer, by default, on the right side only are displayed the entry accessions, but you can show to display the name as well. This will be the short name associated to the entry. And when you hover over each domain, you have a tooltip showing some basic information about the entity. For example, the entry identifier, its short name, its entry type, and the domain. When you click on the domain, it just collapses. So if you want to, you are not interested in the tooltip, can just disable. You can export the data into a different format for further analysis. If you want to see more details, you can zoom into the protein, either by moving the gray bar at the top, or by clicking on the plus and minus button here at the bottom. You can also visualize the protein viewer in full screen mode. Another information I want to mention here, that here at the top you have information about the hydrophobicity properties of each amino acid. Finally, from the CDD and SFLD member databases, we sometimes have site annotation, residue annotation, and this is represented by small rectangles, as you can see here. So if we go back at the top, here on the entries section, you have some information about all the InterPro entries matching this particular protein sequence. And you also have the sequence that you have submitted for the search, which you can reuse to perform another InterProScan analysis by selecting other member databases, for example. And you can also perform a search against the Hammer web server. So let's go back to our presentation. So to summarize, InterProScan provides information about the protein family membership, the different domain architecture, the per residue annotation provided by the CDD and SFLD member databases, the hydrophobicity properties for each residues, and also I forgot to mention, but we also provide GOTERM from the gene ontology on biological process, molecular function, and cellular location. Okay, let's now have a look at more details on the different InterPro pages. So we are going to first explore the browse feature. So this allows you to filter the data based on specific criteria. So let's imagine we are interested in finding the pointer signatures that are integrated in InterPro family type entries and have a GOTERM which is matching a molecular function. So on the InterPro website, this is accessible on the Browse tab here on the top menu. So on the Browse functionality is available for entries, proteins, structures, taxonomy, proteomes, and sets of data. On the left side here, you have you can do a pre-filter on the member database, and here at the top we have other filters. So we said we are interested in finding pointer signatures. So we are looking at the integrated database, which is a type pointer. So we apply the filter. You can see already that the display is adapting based on our selected database. And we want the family entries. Finally, we were looking for the gene ontology terms that are molecular function. So we select this criteria. So we can see that we have about 3,000 InterPro entries that are matching these filters. Those data can be exported in different formats. And here is a table of the different InterPro entries matching with the entry type, the InterPro entry accession, InterPro entry name, the different signatures integrated in this entry, and the Go terms associated. By default, for every table displayed on the website, we only show 20 results at a time, but you can change those settings by clicking on the wheel here on the right side. So we have found about 3,000 signatures that are pointer signatures integrated in InterPro families and have a Go term and molecular function associated. So now let's imagine you are interested in doing a particular text search. You are interested in a particular protein family or a particular domain. So you are going to perform a text search on InterPro. And for this example, I have chosen to look for a protein kinase domain InterPro entry. So we are going to perform a search by text. So in the search section, we are going to search by text this time. You can search by InterPro entry accession, some text, some protein accession, MERV database signatures identifier, Go terms, 3D structures, or proteomes. For more specific results, I recommend to put your text search, if it's letters, words, in double quotes. We are looking for protein kinase domain. So here we have 72 entries matching, but we can see here at the top we have the protein kinase domain, which is an InterPro entry. InterPro entries accession always start with IPR. By clicking on either on the accession or the name, you will access the InterPro entry page corresponding. So let's have a look at this protein kinase domain entry. So here on the left side, we have an inform letter representing the entry type. So here it's a domain, the InterPro entry name, InterPro entry accession. Following, we have a menu with different sections. We have the proteins. So yeah, when an error happens like this, you can reload the page. And hopefully this will solve the problem, like here. Or you can also have some information here on clicking on the three bars at the top, and you can access the services information. So under our proteins, we have the list of proteins matching this InterPro entry. You can see where the InterPro entry matches the particular proteins. And you can filter either by reviewed, unreviewed, or both type of proteins. By clicking on the accession on the name, you can access the protein page corresponding. On the following section, you have the different domain architectures matching this InterPro entry. This is the domain composition of the InterPro entry we are looking at. We also have a section presenting the taxonomy, so all the different species matching this InterPro entry. And below we have a list of the different taxa, which can also be shown as a phylogenetic tree. Each node of the tree is clickable, and then it expands to have more detail. And the following section of the menu shows information about the different proteomes matching this InterPro entry. You can generate a FASTA file corresponding to each of them and export the data for further analysis. On the structures section, we have information about all the 3D structures matching the entry with, on the right side here, the same model as previously. You have the 3D structures and where the InterPro matching is matching the structure. We also have information about protein-protein interactions when available. And pathway, the different proteins matching this InterPro entry are belonging to. This is provided by the KEGG and MetaSync databases. And finally, we have some information about the genome 3D domain predictions. So let's go back to the overview page. Here at the top, we have some information about relationship to other InterPro entries. So relationship to the homologous superfamilies, which is a particular entry type, which contains proteins that share a common evolutionary origin based on the structural features. And we also have information about the relationship with other InterPro entries of the domain type, as we're looking at the domain entry. This can be expanded. And you can click on any of them to go to the particular entry. Here on the right side, we have information about the member databases that are contributing to this particular entry. In this case, we have SMART, PFAM, and ProSite profile signatures. If you want to add more references or more information about this particular entry, you can contact us using this Add Your Annotation button. If we go a bit below in the page, we have general information about this particular InterPro entry, about the functionality, the domain architecture, the species, or any involvement in a disease, anything like that. This is all based on scientific literature. We also provide gene ontology terms annotation when possible. And the references mentioned in the descriptions are from here. We also provide information on cross-references. So for example, here, a link to the ProSite. So to summarize, an InterPro entry page contains information about the InterPro entry type, domain, and accession. The member database signatures contributing to this entry, the different relationship to other InterPro entries, description of the InterPro entry, literature references, gene ontology terms on molecular function, for example, cross-references, and a button to suggest annotation improvements. From the top menu, we have access to the protein matching, this entry, the different domain architectures, the taxonomy, the proteomes, the 3D structures from the PDB, the interactions, the different pathways the proteins integrated in this entry are involved in, and finally, the genome 3D domain predictions. Let's now imagine you are interested in a particular member database signature and you want more information about it. This is possible by accessing the member database page in the InterPro website. I have chosen to look at the example of the coronavirus S2 glycoprotein PFAM family. So this is accessible by entering the signatures accession anywhere in the InterPro website, either in the search by text or from the quick access search here at the top. So on the InterPro member database page, we have information about the name of the signature, its identifier. We have some general information about the signature, the InterPro entry it is integrated in, if it is integrated, and a link to the corresponding member database website. From the top menu here, we have different sections, so we can access the different proteins matching the signature, taxonomy, the proteomes, the different structures, and what I want to look at now is the different sets. So the sets are given by the PFAM, PRSF, CDD, and pointer databases. So in PFAM, they refer to the clans the signature is integrated in. So for PFAM, we also provide the signature. This is based on the logo from Skyline, and it provides information about the probability of each amino acid to be present at a particular residue alongside the sequence. So the bigger the letter is, the more probability there is to find this particular amino acid at this position. So to summarize, the member database page provides general information about this particular signature. We have a link to the member database website and also the InterPro entry it is integrated in. On the top menu section, we have the different protein matching this member database signature. We have taxonomy, proteomes, all the structures. We have the sets from the PFAM, PRSF, CDD, and pointer databases. And we also have amino acid probabilities for each residue. Now let's imagine you are interested in a particular protein accession and you want to find some information about the protein, the 3D structures they are matching. So in my case, I have chosen to look at the protein accession 00308. And so to do so, we're going to go on the InterPro website and search. Well, let's use the quick search here. So we said the protein 00308. Click enter, and then you arrive directly, and because it's a perfect match, to the to the UniProt page corresponding, the protein page corresponding. Here you have some general information about the protein, link to the UniProt. You can download the data in a TSV format. You can choose also to display the different protein isoforms. But what we are more interested in is protein family membership when available. And here we also have again sequence viewer. This displays similar information as the information we had in the sequence viewer as a result of the protein search. But we also provide some, so as you can see, some residue information. But what is more interesting is we provide information about disordered regions. So disordered regions are have little evolutionary conservation, so they are difficult to model as signatures. But this information is useful because those regions have an impact on the flexibility of the protein. So this information allows us to know that, oh, there isn't any domain from an InterPro entry associated to this particular region of the protein, but we can see it is a disordered region. So this gives complementary information on the InterPro annotation. We also provide information from the 3D structures and domain prediction from the Genome 3D partners. This is really useful when there isn't any domain annotated at the InterPro level. This gives complementary information as well. And what is interesting, you can also show the sequence conservation based on the PFAM domains. So the more dark blue the residue is, the more conserved it is. So you can zoom in to have more details for each residue to see which one are more conserved. Let's see. We also provide information on the gene ontology terms on biological process, molecular function, and cellular component when available. If we go back at the top here, we have different sections. You have the entries, where you can have a summary of the InterPro entries this protein is matching. We have access to the different structures. So here at the top, you have sequence viewer again with a summary of all the InterPro entries that are matching this particular protein, and the structures matching as well. So you can see where it is matching the protein alongside the sequence. And below you have detailed information, and you can click on the particular structure to access the InterPro structure page, which provides general information about the structures, the different InterPro entries it is matching by clicking the entry section, and the proteins it is matching. We also have a link to the PDB database. Here you have 3D viewer of the structure, which you can interact with by rotating the molecule, zooming and out. And below we have a sequence viewer again with some information about the chain in the structure, the secondary structure information, and the different InterPro entries matching. When you hover over any part on the sequence viewer, you can see that it is highlighting the molecule in the 3D viewer here. You can obtain the same result by clicking on the menu here. So let's go back to our protein page. And so here on the sequence section, you have the full protein sequence, which you can then use to run against InterProScan or using the Hammer server. We also have a section on similar proteins. This is based on the similar domain architecture. So at the top here, you have a summary of the domain composition of this protein. This is based on PFAM, but you can also display the information based on InterPro accession. And below you have the list of proteins, which you can click on to go to the corresponding protein page. So to summarize, on a protein page, you have general information about the protein. You can also choose to display the different isoforms in the sequence viewer. We have information about the protein family membership, a link to the UniPro database. And from the sequence viewer, we have the similar information that was available as a result of the InterProScan sequence search. on the InterPro entries and member databases mapping this particular protein. We also provide residue annotation, for example, binding pockets. And we have genome 3D domain and structure predictions and information about the sequence conservation. From the top menu, we have access to all the InterPro entries matching this particular protein, the different PDB structures, the sequence of the protein, and similar proteins based on the domain architecture. So now I'm going to show you briefly how you can search on the InterPro for a particular domain architecture. So let's imagine you are interested in proteins with the MH1, MH2 domains. So to do so, we are going to perform a search, but this time not by sequence, not by text, but by domain architecture. So here you can select the domain to include, the domain to exclude. So we said we want the MH1 and MH2 domains. So we select the MH1 and the MH2 domain. So you can see there is 35 different domain architectures that are found with MH1, MH2, and any other domain architecture. But so to make sure that the order of our domains is correct, we're just going to select the order of domain matters option. You can swap the domains if you want to order them in different order. And we only want the ones that have an exact match of those two domains. So we are going to select the exact match option. So we can see that there is 4,447 proteins with this MH1, MH2 architecture. You can click on it to access a list of proteins and export them for further analysis. So we had a total of 4,447 proteins matching this MH1, MH2 domain architecture. So to conclude this webinar, you can use InterPro to identify domains and sites within a new protein sequence and to define its protein family it belongs to. You can also search information about a specific protein, domain, or member database signature from one of the 13 member databases integrated in InterPro. You can also find some information about residues of interest, for example, binding sites or binding pocket and conserved sites in a protein sequence. And from anywhere in the website, all the tables are downloadable for further analysis. To finish, I'd like to thank the InterPro team for their hard work on the curation and the website and production. I'd like to remind you that next week, my colleague Gustavo will present you how you can access InterPro programmatically and how you can download all the data for more analysis. Thank you for your attention, and if you have any question, I'll be happy to answer them.
Thank you, Tiffany, for giving us that tour around InterPro. I see lots of people have been asking questions so far in the chat box, and thank you to Gustavo and Gift for your answers there. But if you do have any further questions, do drop them in and we can ask them now. I see someone's asked about getting a video of the webinar. Yeah, we are recording this, so we'll send out the link to the video tomorrow, and to make sure that everyone who registered will see that. Just before we get to the questions, as Tiffany did say, Gustavo will be back next week for the final of this 3-webinar series, accessing InterPro programmatically. So do register for that and come along and join us next week. If you want to see the other webinars that we have coming up, do go on to our website and check those out. We've got a few listed there at the moment. And also keep an eye on our Twitter @EBI Training to see what we have coming up in the future. And also we let everyone know when the links to recordings go out as well via our Twitter. So I think we have one question here that's come through. Can we search protease substrates based on the domain or sequence? That is.
Protease. I don't think we provide substrate information, but yeah, you can have a go and search for protease and find out what information is available on the website.
Okay, thank you. And the next question, what are the repeats in proteins?
So the repeats are very short regions in the proteins that are repeated multiple times. So it's a motif that is repeated multiple times in the protein.
Okay, great, thank you. And one more that's come through. Can we have a homology structure prediction for unknown protein sequence in InterPro?
So in the sequence search, you have then access to the InterPro entry that is corresponding, and from this InterPro entry, you can then have a look at all the structures matching this particular InterPro entry, and then you will have some information on those structures.
So thank you very much to Fen and to the other members of the InterPro team who have been helping out today. And we hope to see you next week for the Accessing InterPro Programmatically webinar.